Hi all, it is time for our third mini lecture of the digestive system. So we are going to pick back up with the small intestine and then move into the large intestine. I'm going to try and keep this one a little bit shorter since the last one was a little bit long. So let's get right into it. So in the small intestine, we do have intestinal crypts. They are going to be different than the crypts that we saw in the stomach. This time they are going to occur in the floor of the small intestine between two villi. So here we have one villi, here we have another, and then down here we are going to have sort of this pore that opens into our intestinal crypt or into a gland-like structure. In this intestinal crypt, we are going to have enterocytes still, so we're still going to have some absorptive cells. We are also still going to have our goblet cells, which are both cell types that are going to be seen in these villi themselves. We also have stem cells. These stem cells don't necessarily need to be replaced quite as often as the stomach, but we still have a lifespan of 5 or 6 days. And so we shed all of those epithelial cells and we replace them with the stem cells from this intestinal crypt. They are going to, those stem cells will divide. They're sort of usually in the lower region, so they're down towards the base of the crypt. They are going to divide and sort of move their way up and replace the cells of this villi from there. We also are going to have Paneth cells. These are cells that are primarily involved in defense. They are going to secrete lysozyme and defensins, but essentially what we need to know is they secrete compounds that are going to help defend or prevent infection of the body. Overall, our intestinal crypts do secrete intestinal juice. We are down in terms of the volume here. We're at 1 to 2 liters per day. Most of that is going to be in response to, oh, that's a nice font size change there, acid, hypertonic chyme, or distension. So those are the same things that we've already seen the small intestine responding to as we move intestinal contents or stomach contents into the intestine. Primary contents are going to be water, mucus, very, very, very little enzyme. We end up with a pH here of about 7.4 to 7.8. So our pH is up. Again, not a lot of enzyme, mostly just water and mucus. This, in the duodenum, we are also going to see these glands are going to secrete bicarbonate, which is going to help neutralize our stomach acid. The need for that secretion of bicarbonate-rich mucus decreases as we move out of the duodenum, so that's going to be the primary place that we see that particular feature. All right, I realize that I can't play these videos since I have them embedded in our PowerPoint. Since you have access to the PowerPoint, I would encourage you to watch this video. So this video is going to show segmentation within the small intestine, and it's pretty cool to be able to see the actual repeated contractions in the small intestine. Our sort of 3 major purposes for intestinal motility are going to be to mix our chyme, right? And so we have our intestinal juice, which is neutralizing. We have bile and we have pancreatic juice, which are going to help with our digestion process. We need to mix all of those things together. And so we are going to do that via segmentation. So I'm going to write, this is one type of movement. We also want to make sure that our chyme comes in contact with the mucosa of the small intestine. If we don't have contact between the chyme and the mucosa, we're not going to do any absorption. And so that is going to be really, really important. And then the last reason for us to have movements in the intestines is to get rid of any residue by moving it to the large intestine, right? So the first thing we have to do is mix everything together. The next thing we have to do is sort of churn it up and do the rest of our digestion and absorption. The third thing we need to do is move those residues towards the large intestine so that we can eliminate them entirely. And so this slide is a reminder of the difference between segmentation and peristalsis. Up to this point, we have exclusively talked about peristalsis, so I think we probably know what that is, but let's talk about segmentation a little bit. So segmentation is actually our most common type of movement, particularly in the intestines. We are going to get mixing and churning. We have the presence of pacemaker cells. Those pacemaker cells set the contraction pace. Usually we get contractions at about 12 times per minute in the duodenum, and then they reduce as we move down and into the ileum. Once we're in the ileum, we don't need to be moving as much because we're not doing digestion and absorption to the same level. When the nutrients have been absorbed, which again is as we move towards that ileum, we reduce the rate of segmentation. Segmentation itself is going to be movement of stationary ring-like constrictions. So we're going to get, oops. You can envision this like you're squeezing a tube of toothpaste. You start by squeezing it at two places just randomly, okay? And so when you do that, the contents are going to move to one direction or the other. Then you can move your fingers down and squeeze at two different places. And again, we're going to get movement of the contents where you're squeezing to out to either side. So as you do this sort of repeatedly, time and time again, just moving where you're squeezing or making those rings of constriction, you're going to end up mixing the contents of that tube into sort of a uniform substance. And so that's one of those reasons or one of those purposes of segmentation. We do still get peristalsis in the small intestine. With peristalsis in the small intestine, the major purpose is going to be movement of any residue towards the large intestine, so propulsion from place to place. This happens often as the gastroileal reflex. So when you put food into your stomach, it tells the ileum that it actually needs to move contents out of the small intestine so the small intestine has room for the new food that's going to be moving out of the stomach. Right? So gastroileal adds stuff to the stomach, small intestine has to make room. So when that happens, we get an increase in segmentation as well as peristalsis, and we move all of the contents of the small intestine into the cecum by using that peristalsis-like movement. So we kind of squeeze from one end of the tube to the other and progress it from place to place. Speaking of the large intestine, let's quickly go through what's happening here. At this point, we've already digested and absorbed pretty much everything of importance out of what we've eaten. So the large intestine is going to get the leftovers. So it gets the things that we can't digest, it gets the residues. We're essentially going to be doing compaction and elimination in the large intestine. In total, over the course of a day, we see about 500 milliliters of stuff that we can't digest come into the large intestine. It reduces that volume to about 150 milliliters, which is eliminated by defecation or through feces. The reduction from 500 to 150 milliliters is mostly accomplished through absorbing of our water. And it is going to begin down here in the cecum. So here's our small intestine, that's the ileum. That's going to be our ileocecal valve right there. Contents from the small intestine are going to dump into the cecum. The cecum is actually inferior to that opening, so it's down here. Off the end of the cecum, we have our appendix. Right? So the appendix is sort of this pinky-like projection off of the inferior end of the cecum. It's a reservoir for some of our lymphatic tissues. Lymphatic cells rather. From the cecum then, so we'll sort of fill that area, we're going to enter into the colon. The colon has 4 components. So it goes into the ascending colon, ascending because it moves up the body or it ascends. It then flexes over and forms the transverse colon, which goes from the right side of the body towards the left side of the body. So all the way from one side to the other. Contents will then move down the descending colon. That's going to be down the left side of the body towards the sigmoid colon. So the sigmoid colon forms this S shape. That's here. Sorry, I meant to be highlighting these. The sigmoid colon is then going to empty into the rectum. The rectum then goes out through the anus. In terms of our other structures, here we can see sort of all of those things I just pointed out. Here's our ileocecal valve, right? So the opening from the small intestine into the large intestine. There's our cecum and our appendix. Ascending colon, transverse colon, descending colon, sigmoid colon into the rectum, and then out the anus through the anal canal. Just like our other organs in the digestive system, we have our standard sort of 4 layers. We do have a modification here. So our muscularis externa now actually forms what are called tinnae colae. These are longitudinal fibers of thick ribbon-like muscle that are evident sort of on the external surface. So we can see these here all the way down. We get these 3 bands. When those are tightened, they form these what are called haustra or pouching. And that's because we have this muscle tone that pulls and causes sort of these little pouches to form. We also often have the presence of omental appendages, which are kind of these little fatty pouches of the peritoneum. They are also called epiglottic appendages. So you'll see these sort of little pieces of fat basically just hanging off the large intestine. Their function is unknown, so we don't really know what they're there for, but they are very common. And again, we have lots of lymphocytes here because we have quite a large bacterial population. We need to make sure that we are protecting this tissue and the tissues that surround it from any cells that may or may not get out of this particular organ. As we move out of the colon, we actually move into the rectum and then into the anal canal. So the anal canal is going to have 3 curves and 3 infoldings, which are called rectal valves. The purpose of those infoldings and valves, and so here's an example. Here's our rectum. So we'll have feces moving down into the rectum. We then have these rectal valves, and there are usually 3 of them. They are going to help us retain feces when we are not ready to defecate. So it's a mechanism that helps us sort of hold feces in the rectum so that it doesn't just fall out, which is a lovely image. In the anal canal, we have the presence of our hemorrhoidal veins. So these hemorrhoidal veins are very superficial. They are subject to a lot of pressure from above. So as body weight or as feces fills the rectum and pushes on these veins, they sometimes rupture through and then come out through the anus, right? And so that would be the presence of a hemorrhoid. They can be removed. They sometimes shrink and sort of go back up in a little bit, but they're never going to be sort of fully encased in this tissue again. So our anal canal is going to pass through some skeletal muscles. It is going to be closed off by actually 2 anal sphincters. So let me again highlight these. So we are going to have an internal anal sphincter and an external anal sphincter. So we have 2 different muscle groups here that are controlling exit of feces out of the anal canal. The internal anal sphincters are going to be composed of smooth muscle, hopefully you remember that our smooth muscle is involuntarily controlled. So we do not control when this internal sphincter opens or closes. The other sphincter is external. Fortunately for us, it is composed of skeletal muscle, which means that we can control it. This external anal sphincter made of skeletal muscle is what allows us to determine whether now is an appropriate time to poop or not. Okay, so we are very thankful that we have, actually both of those are very useful to us, but we are particularly grateful for the fact that we have this skeletal muscle anal sphincter. And then other things just to mention quickly are our anal columns and sinuses. So these anal columns and sinuses are going to produce mucus. That mucus is going to act as a lubricant, again, during defecation. Oftentimes our feces is pretty dry. We've absorbed a lot of water out of it, and we don't want to damage this region a lot by pushing something really dry and sort of rough through. So we sort of coat it up with mucus. We make sure that the feces moves out very easily and does not actually damage the tissue that is here. Okay, so this will be the last slide I talk about here, and again, I'll sort of pick back up on this in our next mini lecture. The mucosa of the large intestine, we're back to our very simple columnar epithelium, which we've seen in most places throughout the digestive system anyways. In the anal canal, we do change that, so it becomes non-keratinized stratified squamous epithelium. We have stratified squamous epithelium where we want extra durability and friction resistance. We don't want our anal canal to be damaged by the feces that's present. So we switch to stratified squamous there to prevent that. We do lack the folds that we see in the small intestine, so we're not doing any more absorbing here, so we don't really need those folds. And we do maintain, however, intestinal crypts that are primarily for mucus production. So not really a ton to talk about in terms of microscopic anatomy of our large intestine, because it's primarily just sort of absorbing and compacting our residue. Okay, so this is the end of our third mini lecture, and I will pick up again in our next one. Hopefully things are going well. Please let me know if they are not, and I look forward to talking to you guys again through my computer.